In this episode, we're going to talk about resting membrane potential. I'd like to tackle the last term first. Potential. What does that mean? If you have potential, it means that you can do something, but you haven't done anything yet. Batteries have potential. They have potential energy, and that's measured in voltage. We know that batteries store that. They have a certain amount of potential energy. If we were to look at that battery's voltage, it's a measure of its potential energy over time. It's measured in volts, and this battery has 1.5 volts in it. And we know that as we use that battery and we use the energy over time, it goes dead and it goes down to 0, and then we take it to the hazardous waste and it's done, unless you have rechargeable batteries. Well, the cell is going to operate in a somewhat similar fashion, but here we're measuring the membrane potential. So here's our cell again, and we know that the cell membrane separates the inside and the outside environment, and that's where we're going to measure that potential energy across this membrane. How does the inside and outside compare? What does the potential energy look like across those? And we can measure this voltage as well across the membrane. In this case, however, our voltage is smaller. We're measuring it in millivolts, so 1,000th of a volt. And the scale is a little stranger. 0, instead of being at the bottom, is here. And our cells typically want to keep their voltage, their membrane potential, around -70. Not every cell, but most cells keep it around that. Now, cells are constantly spending energy and receiving energy, and so that usually stays fairly steady over time. Some ups and downs, concept of homeostasis. And we don't want our potential to get to 0. The negative's a little bit strange, but we'll work on that. The idea is we keep that membrane potential fairly steady most of the time, not going to 0. And let's see how that is done. So there's a few aspects that contribute to that resting potential of -70. One of those is simply the proteins, stuff in vesicles and ribosomes, the mitochondria, other proteins, free-floating ribosomes around the cell. There's some inherent negative charges there, and so that gets us partly negative charge. The inside of the cell is more negative than the outside. So if you just compare the two, the outside would have a slightly positive charge compared to the inside of the cell. So that gives us a little bit of potential. Definitely not 0, but we're only a little ways down. So it doesn't get us all the way to -70. We're going to have to call in some other factors in here when we get to that. And most of those have to do with two familiar ions, sodium and potassium. I'm going to show sodium as red beads, potassium as orange beads, and they're going to need a way to get in and out of the cell. So we know that the membrane is selectively permeable. I'm going to make it permeable to potassium by putting in potassium-specific leak channels. So in these channels, if you had potassium in the cell, it can leave through those channels, it can come in through those channels. It's not one way. It can just go either direction as long as there's a leak channel, but it can't get through the rest of the membrane because that membrane is made of phospholipids, which would repel the potassium ion. For sodium, we have some sodium leak channels as well. Cover up these other ones with that, and same thing. If you had sodium in the cell, it could leak out, leak in, all using facilitated diffusion, going down their concentration gradient and basically spreading out. Well, that doesn't seem like that's building much potential energy, and you're absolutely right. we need to spend energy using the sodium and potassium pump. That is another membrane protein that uses ATP. So it uses ATP and breaks it down into ADP. It's the mitochondria that make the ATP and supply the sodium-potassium pump with the energy that it needs. Let's start with a few more of these sodiums all over the place. Let them run everywhere. That's what they do in the body. They're kind of unwieldy. Some outside, some inside. And the same with potassium. So now we have a good mix of sodium and potassium. They're spread out all over the place. If we go back to our voltage, we're really closer to zero, other than our negative charge ions inside. The number of positive ions outside and inside are essentially equal. Remember, sodium has a positive charge of +1 and potassium of +1. So essentially the pluses outside equal the pluses inside. There's no potential energy there. So let's start spending some energy and building up some potential. So we'll activate our sodium-potassium pump using ATP. And the sodium-potassium pump has 3 slots for sodium, and it has 2 slots for potassium. And so for every ATP that we spend, we're going to put those on a little merry-go-round. The potassium comes in and the sodium goes out. Notice that 3 went out and 2 came in. So if 3 positives leave and 2 positives come in, and you do a little quick math, then you realize that basically the inside has a -1 charge, or 1 minus. It's more negatively charged. And so now we're building up a little bit of potential energy. We're starting to get more negative, further away from zero. Well, if we keep operating the sodium-potassium pump, then sodium goes out, 2 potassiums come in, 3 sodiums go out, 2 potassiums come in, 3 sodiums go out, 2 potassiums come in, and so on and so forth. It's starting to get a little sparse. You'll notice, at least over in this area, let's get rid of these guys. 3 go out, and 2 potassium come in. Leaving aside all of this mess over here, let's just take it off and clean this up a little bit. I can see that I've built up a difference between the outside and the inside. That sure looks like potential energy to me. We have a high concentration of potassium here and a high concentration of sodium out here. Sodium now wants to come in. It can't come in through any part of the membrane. It can come in through the leak channels. But if it did, then it would be pumped right back out and more potassium would be brought in. So that's responsible for the big part of our potential energy. Now we're down, getting close to -70. We're not quite there yet. By spending some energy and pumping sodium and potassium out, but always with more positive ions leaving the cell. All right, so we have 2 factors. There's really one more. Keep it in mind, this membrane is a very important player in keeping the different ions on either side of it. We have the proteins and other molecules that can't leave inside that are negatively charged. We have the sodium-potassium pump, and now I want to address the leak channels. The number of those is important. There are more potassium leak channels than there are sodium leak channels in most cells. And what happens, let's allow these, let's just line them up, assuming that potassium is going to leave down its concentration gradient by facilitated diffusion with these channels facilitating, and sodium would want to diffuse in. Well, here let me take 1, 2 sodiums coming in to 1, 2, 3, 4, 5, 6 potassiums going out. Well, let's do the math again. That's 6 positive ions leaving with only 2 positive ions coming in. Difference there is 4. So I have 4 charges difference, with the inside lacking those positive charges. So the inside becomes more negative. And that gets us on our graph down into this negative 70 range that we want to stay at. And there's going to be some play when sodium comes in. Now you're adding more positive charges to the inside, and so on the membrane potential you're going to go up a little bit toward the positive, but then the sodium-potassium pump will operate and it'll go back down. So there are some ups and downs, but the point is the cell now has potential energy, and it can use that to do work. It can use this sodium gradient to drive secondary active transport. It can line up voltage-sensitive proteins and send electrical signals by allowing sodium in and potassium out. That's called an action potential. But the cell now has potential energy thanks to the sodium-potassium pump, the difference in potassium and sodium lead channels, and some of the negatively charged larger proteins within the cell.